Hello, can anyone hear me? Initially, when I logged in, nobody else logged in, and I thought that was rather strange. So I closed it down and restarted it, and a bunch of people started logging in once I restarted it. Um, did anyone else have that problem? All right. If not, let's go ahead and let's get started. Are there any questions before we get started? Am I sharing my screen? Not hearing
No,
anything. Thank you. All right. So today is October 5th, and we're supposed to be on chapter 2. We don't have to finish chapter 2 today, so we should be good. There will be a lab in the lab time today. Any questions about the schedule? All right. Oh, where do I have that? Didn't open it. Before I begin, let me make sure that I've started. Guess I should leave that one. We're finished with chapter 1 and chapter 10 now, so I want to make sure you guys have access to the notes, to my notes to chapter 1 and 10, meaning the slideshow. If ever I don't have them available after I've finished the lesson, you can send me an email to remind me. And I don't think I did, but let me check right now. Boy, this is awfully slow today. There you go. Let me see if there's anything else I need to talk about on the schedule. Hopefully, you took the practice quiz. If you did not, and you want to, I think you still can. Let me check on that. Yeah, you have until 11:59 p.m. tonight to take the practice quiz. I really recommend you do for two reasons. And that is first, to get a little bit more familiar with my quizzes. And second, you need to get Respondus Lockdown Browser downloaded on your computer and up and running. Because if you run into a problem, especially if you're late in the day, you'll need to contact the computer help desk. Unless you're brilliant, you can change the settings yourself. And then they'll help you change the settings to get it up and running. I've never had a problem with Respondus Lockdown Browser working on a student's computer. It is true there was one student who didn't get it running. However, she wasn't following my directions. So I have a feeling she wasn't following the computer help desk directions. And she's the only student I never got it up and running on her computer. All right, let's make sure I'm recording this. There it is. So we had talked about water being a good solvent. Let me make sure I don't have another marker. Usually I don't put the 2 here or there. I guess I did put it there. That's an unusual place for me to put it. Normally I put it on the what do you call it? The title. All right, so we talked about water being a good solvent. Water is an inorganic compound, and it does hydrogen bond between other water molecules. Because of the hydrogen bonds between water molecules, water is excellent at buffering the temperature. Water can absorb a lot of heat before it becomes heated or evaporates, meaning that when water absorbs the heat, some of that energy is taken to break the hydrogen bonds between the water molecules. The other energy is taken by the water molecule itself, causing the water molecule to vibrate and move more rapidly. Furthermore, when the hydrogen bonds break off from a water molecule, the water molecule is not going to stay in the liquid, and then that molecule will evaporate. So that means the molecules of water that have the highest energy, or you can say, since their amount of energy is their temperature, it means that the water molecules with the highest temperature will be the ones leaving, and that will keep the rest of the water cooler. Any questions about that? So as the hydrogen bonds break, the water molecules break off from the liquid water and evaporate, taking the heat away with it. Now, water can absorb energy in two ways. The energy can be absorbed by the molecule, and then the molecule will move about more rapidly, and that is a measure of the temperature of the object. All molecules, all objects, can have a temperature, and it's simply the speed that their molecules are moving. Water can also take the energy to break off the hydrogen bonds. Other substances do not form hydrogen bonds, like oil. And therefore, if you were to give the same amount of energy to water and to oil, the oil will get a higher temperature, or the temperature will rise further than water. Any questions about any of that? Another thing that water can do is that water, H2O, can split into H plus and OH minus. This is on the surface of the Earth at normal conditions. The H2O can split into H plus ions and OH ions. We call the H plus ion a proton, because that's what a proton is, and the OH minus of the hydroxide ion. These charged molecules in water participate in chemical reactions, and they also give the water's pH. Now, you can break H2O into two H plus ions, which are plus, and then a one oxygen ion with a -2 charge. No. However, that does not happen at the temperature of the Earth at normal conditions. It does happen at the surface of the sun, but that's quite a bit hotter than the temperature of the Earth. Okay, so water does not split into two H pluses and O minus, and it's a 2 minus, on the normal conditions on the planet Earth. Any question about any of that? You notice how I said normal conditions. You could put electricity into the water, and that energy would actually split the H2O into 2H pluses and one O with a -2 charge. And that we call electrolysis. But that's not a normal condition under the planet Earth. That's a man-made condition. All right, let's talk a little bit more about the pH. pH gives water its pH, or its acidity, or you could say its alkalinity. And there are different pHs. They range from pH 0, being very acidic, to pH, oops, not small what I wanted. Close that down. Trying to blow this up. To pH 14, very basic. At neutral pH, which is pH 7, one water molecule splits into H plus and OH minus, and the concentration of H plus and the concentration of OH minus in the water are the same. So that's neutral water at pH 7. At lower pH numbers, we have increasing acidity, such as urine being pH 6 or around that, wine and tomato juice being around pH 4, lemon juice being around pH 2, and then stomach acid being around pH 1.5. The most acidic thing that you're probably familiar with around pH 1 is the acid in your car battery. And that's the most acidic thing that I know of. Anything, questions about that part? Now, seawater is around pH 8.5. That's slightly alkaline. Human blood, I can say, is really slightly alkaline. It's between pH 7.2 and pH 7.4, very slightly alkaline. Seawater is slightly alkaline. Milk of magnesia around pH 10.5. Household ammonia around 11.5. Bleach, household bleach around pH 12.5. And probably the most alkaline thing that you might be familiar with is oven cleaner around pH 13.5. Most of you have probably never come across lime water. I think it's used in industry. I've actually never used lime water, but it's the highest pH you can have at around pH 14. So we call the numbers higher than pH 7 basic or alkaline. When you have increasing acidity, you have more H plus ions in solution than you have OH minus ions. With increasing basic conditions, you have more OH minus ions in solution than you have H plus ions in solution. Any question about any of that? All right, let me see where I talk about pH. I don't, so I'm going to talk about it here. Oh, there it is. Sorry, found it there. It's the last slide here. So we'll wait on that and then just move on, talking about acid, bases, and salts. The textbook will tell you that a substance that dissociates and gives off one or more hydrogen plus one ions is an acid, like HCl. When it dissolves in water, will become H plus 1. And you might notice that we abbreviate the plus 1 as simply a plus. That's a complete plus 1 charge. And then it separates into the chloride minus 1 ion, because there's H plus being given off by HCl, it is an acid. And you put HCl in water, we'll get this acid solution here with H plus being higher in concentration than the OH minus in the water. The textbook will tell you that a base is a substance that dissociates, giving off one or more OH minus ions. And that's an OH with a -1 charge. Like sodium hydroxide, when you put it in water, will dissociate into sodium +1 and OH -1. So that will increase the OH -1 in solution, and that will be a base. Any question about that? Now, the textbook does not tell you that a substance is a base if it gives up OH minus or it absorbs H plus ions. For example, bicarbonate, like in sodium bicarbonate, that's baking soda in our kitchens. You put that in water, it will absorb an H plus in the water, making bicarbonate, H2CO3. So sodium bicarbonate is a base because it doesn't give off OH minus, but it absorbs H plus ions. And I don't know why your textbook author does not discuss that, but a base is something that either gives up OH minus or absorbs H plus ions in solution. Any question about that? Okay. By the way, bicarbonate is an important buffer in your blood, helping neutralize the pH change in your body and in your blood. So we have, this is one of our main buffers, bicarbonate, and then carbonic acid. And if you, let's see, if you start eating something that is acidic or drinking a lot of soda, which is acidic, you'll put H plus ions in your blood. And it could be anything else you're eating that's acidic, like lemons or lemonade. It's acidic. And it doesn't harm us because the H plus will bind with the bicarbonate in our blood, forming H2CO3. And then the H2CO3 will be transported by the lungs to, by the blood to the lungs. And then the H2CO3 can dissociate into water in CO2, and then we'll breathe out the CO2, and that neutralizes the acid in our body. Conversely, if you have a bunch of OH minus in your meal, there are some vegetables which are basic, and if you eat a whole bunch of them, it would be neutralized by, let me see, bicarbonate, and this reaction can reverse, which will release the OH plus, and then the H plus will combine with the OH minus, forming water, and that will remove the OH minus from our blood. This very rarely happens. If you really were to get too much OH minus in your blood, this could also dissociate, meaning give off an H plus, which would then bind with the OH minus. But that doesn't normally happen in a person because the pH of the blood would be so far off that the person would be sick and in danger of dying. But that could happen if it needed to. All right, did I talk about everything there? Oh, let's talk about bases and salts. A salt is a substance that dissociates into a cation, that is a ion having a positive charge or maybe more than one positive charge, and it also dissociates into an anion having a negative charge. neither of which are H plus or OH minus. Because if it dissociates into H plus, it'll be called an acid. And if it dissociates into OH minus, it'll be called a base. But if it dissociates into another cation or another anion, we then call it a salt. Sodium chloride, which is table salt, dissociates into a cation, sodium plus 1, and an anion, chloride minus 1. So sodium chloride is a salt. However, chemists will tell you that so is magnesium chloride, a salt, and let me think, magnesium sulfate is a salt. I'm drawing a blank. Magnesium carbonate is a salt. Trying to think of some other things. So there's a whole bunch of substances that can dissociate into cations and anions, and we call them salts, much more than just sodium chloride, which is table salt. Any question about any of that? All right. An electrolyte is a substance that dissociates into water, forming ions, such as sodium chloride. An electrolyte, when it dissociates into ions in water, it'll allow electricity to travel through the water, meaning there's molecules with a charge in the water, sodium chloride, and so salt water or sodium chloride water will carry an electric charge much better than pure water, which would have very few ions in the pure water. And if you don't know, in an electric storm, you should get out of the ocean or any saltwater lake. But if you're in fresh water, it's not something that you immediately have to get out of the fresh water. And that's because if lightning strikes anywhere near you, and I'm talking about several feet or maybe even several yards away from you, you will get an electric shock in salt water. Whereas in fresh water, the lightning has to be relatively close to you before you would get a shock. And how close it has to be is a couple of feet. I don't know if it's 6 feet away or something like that, okay? But it's a couple of feet. It might be 3 feet away or 6 feet away, meaning fresh water does have some ions in it, so it can carry an electric charge. But if ever you're in salt water during a lightning storm, you should get out. Any question about any of that? Oh, here I'm talking about organisms need to maintain a fairly constant balance between their acids and their bases in their body to remain healthy. The pH is a measure of the H plus ions in solution. The exact measure of the pH is the pH equals the negative log of the concentration of H plus ions in solution. I know that's a bit complex, but do you guys understand that? So all you have to do is measure the H plus ions in solution, and then you can get the pH, although you have to take a log of that measure of the H plus ions in solution. Now let's talk a little bit about what pH of 7 means. This means that for every molecule of water that splits into H plus ions, oops, didn't do that right. And obviously there will be an OH minus also. But for every molecule of water that splits into an H plus ion, there are 1 times 10 to the 7th water molecules. And let me go superscript there. Can you guys see this? I'll blow this up in just a minute.
Yeah.
I'm not seeing where that is there. There it is. Oh, there, that's a little better. Make it really big. So a pH 7 means that one water molecule will split into H plus. Let me superscript that. For every 1 times 10 to the 7th water molecules. For pH 6, oops. There will be one water molecule that'll split into H plus ions per 10 to the 6 water molecules. For pH 5, oh, come on. There is one water molecule that splits into H plus ion for every 1 times 10 to the 5th water molecules. So this is the meaning of the pH scale. And then that's true for pH 8 also, and anything higher. One water molecule, oops, got a 5 in there. One water molecule splits into H plus ions for every 1 times 10 to the 8th water molecules. Any question about that? So that's the meaning of the pH scale. And you can notice that the pH scale, a change of 1 pH, is a change in the H plus ion of 10 full. All right, I've already mentioned that an acidic solution is where the concentration of H plus ions in solution is greater than the concentration of OH minus ions, and a basic or alkaline solution is when the concentration of OH minus is greater than the concentration of H plus ions. Most organisms grow best around pH 7. So you could say that most bacteria grow best between pH 6.5 and 7.5. 6.5 and 7.5, yeah, that's right. One other thing I wanted to mention about the pH, if I can remember. Oh, pH of 0. Let me demonstrate what that means. Oh, I already shut that down, darn it. A pH of 0 literally means, let me build that up. That one water molecule splits into H plus ions or 1 times 10 to the 0. Does anyone know what 1 times 10 to the 0 is? Nobody knows? You should know that from your math. 1 times 10 to the 0. Oops, I didn't draw that right. This must be a 1. So 1 water molecule splits into 1 H plus ions per 1 times 10 to the 0 water molecules. And I got to shrink that. What this means is that 1 water molecule splits into H plus ions, and then obviously the OH minus. Do I have that there? Yes, I do. For one water molecule, meaning one will split into, one will split into H plus, meaning everything split into H plus. So that's what a pH of, oops, that's what it's supposed to do. Here we go. That's where the 0 came from. Any question about that? So a pH of 0 is meaning that one water molecule is going to split apart into H plus ions. I'm not sure what the pH of 14 means, but the pH of 14 is as high as the pH can go, and I don't know the meaning behind a pH of 14. Sorry, I'm not a chemist. A chemist could probably explain it to you. All right, let's move on to talking about organic compounds. We're looking at some organic compounds here. And you can see that organic compounds always contain carbon and hydrogen. They may contain other elements as well, particularly oxygen, like this one has oxygen and there's oxygen there and there. But most of the atoms are carbon and hydrogen. Organic compounds can be complex, like this one here, and there's a few of them that can be fairly simple, like methane or ethane. And that's fairly simple. But these are very complex, this one here and that one there. You can draw a chemical compound with the stick figure like this, or you can draw it with the ring figure like this. Let me blow this up a little. When you're using the ring figure, wherever there is a point, that means there is a carbon at that point. So all of these points have carbons. If ever there's not a carbon at that point, I've got that somewhere. Then you have to draw in the other atom. Let me show you that. I think I have it with the glucose someplace down here. There we go. So here's a ring structure, and wherever there's a point, there's a carbon. Wherever there's not a carbon, like there's an oxygen here, you have to put in the O. Any question about any of that? Let me shrink that down now. Anyways, organic compounds can be much more complex than an inorganic compound. Inorganic compounds tend to be fairly simple, like methane, which is not an inorganic compound. But two inorganic compounds we'll talk about in just a little bit are carbon dioxide and water, and they're fairly simple compared to this. So organic compounds always contain carbon and hydrogen. Many organic compounds also contain oxygen, but not all of them. Some organic compounds that are familiar to you are amino acids, carbohydrates, nucleotides, lipids, proteins, and any hydrocarbon. If an organic compound only contains carbon and hydrogen, we call it a hydrocarbon. But like I said, many of them contain oxygen, such as all the amino acids and the carbohydrates and the nucleotides. All the biological molecules contain oxygen in the organic compound. Many of their organic compounds contain other elements like nitrogen, found in protein and in nucleic acids, sulfur, found in, I used to know this, certainly protein, phosphate, found in ATP and DNA and RNA. All of these elements are held together covalently in an organic compound. Usually, most of the bonds in an organic compound are a carbon bond. So the carbon is basically the backbone. Oops, went too far. The backbone. What did I do with that? Of the molecule. So most of the bonds covalently held together in this molecule are carbon. There are a few exceptions, like there's an oxygen. Even here, there's an oxygen helping to hold the molecule together. But most of them are carbon-carbon bonds. Carbon is important in organic compounds because carbon can bind to 4 other elements or atoms. Compared to an inorganic molecule, organic compounds are more complex and they're capable of more complicated biological functions. As I stated, some examples of an organic compound would be all proteins, all carbohydrates, and all lipids. Shrink that down a little bit. Carbon is important to organic compounds because carbon can bind up to 4 other atoms. Carbon atoms can be found bound together in chains or ring structures. So this would be a chain, and then the ring structure would be here. Yep, I think I already talked about that. So carbon tends to be the skeleton or the backbone of the organic molecule. The organic molecule can have functional groups, and this would be a group bound to a carbon, which gives the molecule properties, different properties. For example, methanol, which is methane. Where do I have methane? Right here, which is an organic compound, but one H atom is removed from methane to get methanol, which has an OH group on it, which is a functional group that changes the properties of this molecule compared to methane and gives this molecule different properties. So many of the properties of organic compounds come from functional groups. Any question about any of that? Oh, come on. So functional groups are responsible for most of the chemical and physical properties of a particular organic compound or molecule. Hydroxyl, the OH group, is an alcohol. So you find it in alcohols. And one of the properties it changes in the molecule is methane is not soluble in water. So it is hydrophobic. Whereas methanol, containing the OH group, is dissolvable in water. So it is hydrophilic, meaning methanol attracts water molecules to it and helps it dissolve in the water. The different functional groups that you must know are in the red boxes, like the OH group found in alcohol, lipids, and carbohydrates. It's also found in nucleic acids and proteins. Another functional group is NH2, which we call the amino group, and it's found in proteins. Another functional group you must know is COOH or COOH. And you'll notice that one of the O's has a double bond to carbon. Let me blow that up. The other OH, a single bond to carbon. This is called the carboxyl group, which is found in proteins, lipids, and organic acids. So mainly proteins and lipids. And then another functional group is phosphate, PO4 with a -2 charge. And this is found in DNA and RNA, as well as ATP. Why do you think I want you to be familiar with the functional groups in a red box and not the other groups, like an ester or an ether? No one has a guess? Well, we're mostly going to be talking about biological molecules in this class. And biological molecules have these functional groups shown in the red boxes. Frequently, you can have a molecule with more than one functional group, such as an amino acid, which has both the amino group and the carboxyl group on the amino acid. And we'll talk more about amino acids when we talk about proteins. Any question about any of this?
Can you go back just to the previous slide so I can see what that NH2? Okay, amino, thank you.
Amino group. Okay, you should know the alcohol, the amino group, the carboxyl group, and the phosphate group. All right, organic compounds can be very large, and we call those macromolecules. And if the macromolecule is made up of small organic molecules, we then call that macromolecule a polymer. And a polymer simply means many mers or many units, monomers. And the small repeating units of the polymer we call a monomer. For example, let me see if I got it there. For example, one polymer is starch, and it's composed of one monomer, and that monomer is glucose. And all starch is a bunch of glucoses hooked together to make the starch. So starch is both a macromolecule and a polymer, and it's composed of monomers that are glucose. How you make a polymer is you just take two monomers, or one monomer and a smaller polymer, and then link them together by dehydration synthesis, where the H comes off of this side of the monomer and the OH minus comes off of this monomer, and that'll form water. So water is given off by this reaction. And then the R group here will be bound to the R group there. And so we have RR, or the polymer. This is called a condensation reaction or a dehydration synthesis reaction. Dehydration synthesis because water is coming off and given off. Condensation, I think that's referring to the condensation of water. So water is made. to make the polymer. And then you can reverse that, take the polymer, add water, and go the other way, which you would most of the time make a monomer. The water will split off the H2O with the OH going to this R group of the monomer, and then the H of the water going to the R group here, which most of the time that'll be a smaller polymer. But if this is not a polymer, but a what do you call a bimer? It doesn't sound right. Two monomers hooked together. You can make, split it into two monomers here. And you could have that if that was two monomers hooked together. And I'm trying to remember, there's a name for that. A dimer. There we go, a dimer. This reaction is called hydrolysis, where water is added, breaks the bond, the chemical bond in the R being held together. And although you might not be familiar with hydrolysis reaction, I'm sure you've heard of the hydrolysis reaction because Marty McFly said, "Ma, you sure know how to hydrate a pizza," when her mother took this hockey puck-like thing and put it into her hydrolysis machine, and lo and behold, out came this beautiful pizza. You remember that? That was in Back to the Future too, I think. Anyways, that's how Marty McFly's mother, I don't remember her name, hydrolyzed the pizza. So dehydration is the mechanism for synthesis of macromolecules, such as carbohydrates, lipids, and proteins. And then hydrolysis is the reverse of that, breaking down the polymer into the monomers. Specifically, with dehydration synthesis, you can have a polymer and the monomer can be added. The OH comes off the monomer, the H off of the polymer, and then the monomer can covalently bind to the polymer, making a larger polymer. And then water is given off. Conversely, water can come in to a polymer. Water will split with the OH group of the water joining the monomer, and then the monomer can come off, and then the H of the water will bind to the shorter polymer. And by the way, excuse me, this is how our blood glucose is released from our liver. Glycogen will have water come in and split off the glucose, and then the glucose will be released into the blood. That's how our liver works. And of course, if you eat a very high glucose meal, you will have the glucose being added to the glycogen. And that will be a dehydration synthesis. All right, any question about dehydration synthesis or hydrolysis? So if not, let's talk about carbohydrates. They include sugar, starch, and complex carbohydrates, which starch is a complex carbohydrate, but it also contains other complex carbohydrates. Carbohydrates are an important energy source for organisms. They're important for an organism's cellular and tissue structure. Carbohydrates are important in nucleic acids. Carbohydrates consist of carbon, hydrogen, and oxygen, but it's not just any arrangement of carbon, hydrogen, and oxygen. Carbohydrates have the general formula: for every one carbon, there are two hydrogens and one oxygen. And then you just repeat that for the larger carbohydrate. For example, glucose would be CH2 O in parentheses and then put a 6 for the N. And that would be glucose because glucose is C6H12O6. Carbohydrates are simple sugars or strings of simple sugars linked together. And we can classify carbohydrates into 3 major groups on the basis of their size. A monosaccharide is a simple carbohydrate with only one monomer. A disaccharide would be 2 monomers hooked together. And then a polysaccharide would be many monomers of a carbohydrate put together. And saccharide means sugar, so mono means 1 sugar, disaccharide 2 sugars, polysaccharide many sugars. The monosaccharides are also called simple sugars. They always contain 3 to 7 carbon atoms, but usually monosaccharides contain 5 or 6 carbons. Monosaccharides are sweet and they all are water soluble, or we could call them hydrophilic. Disaccharides are formed by joining 2 monosaccharides together in dehydration synthesis, such as we can combine glucose, shown here, with fructose, shown here, and you notice both of these are C6H12O6, fructose being C6H12O6 also, and then the water can come out by dehydration synthesis to make sucrose, also called table sugar, which is here. And then water is given off. So sucrose is a disaccharide, and then glucose and fructose are monosaccharides. You can do the reverse in a hydrolysis reaction where you have sucrose and water is added, and you can break it down into glucose and fructose, the 2 monosaccharides. Okay, polysaccharides consist of many monosaccharides. They can be straight or branched chain molecules. Some examples of a polysaccharide are starch, shown here, where all you have is glucose, one after the other, mostly hooked together in a single non-branching chain. And I'm not going to complicate that a little further, but you could have a few branches in starch, but not many. Glycogen, which is the way we store glucose in an animal, is similar to starch. It's just that it is a branching chain. So it has more branches in it than starch. And then here are some starchy foods shown here. Everyone knows bread, cereals, and pasta. Rice and potatoes contain a lot of starch, but many people do not know that beans and chestnuts are high in starch. In fact, beans are about 1/3 of their weight is carbohydrates. So they are fairly high in starch. It's just that they're also high in fat and protein, meaning beans are about 1/3 carbohydrate, 1/3 protein, and 1/3 lipids or fats. So they're also high in other things. Okay, but they are, and chestnuts, I don't know the exact breakdown, but they are high on carbohydrates. I suspect they're probably pretty good in protein too. And I don't know the chemical makeup of chestnuts, so I'm not going to even comment on the lipids. Here we're looking at a slide showing you some of the different carbohydrates. You do not need to memorize this slide. You should know starch, glycogen, and cellulose are all carbohydrates, complex carbohydrates. Starch being mostly a single strand of glucose hooked together, one after the other. Glycogen is the same, it's just that it's a chain that branches more often. Cellulose is also a chain of glucose hooked together, one after the other. So here we have cellulose, and what it is glucose hooked together, in the blue lines being the chemically covalent bond. And so all of these rows are glucose held together by a chemically covalent bond. And cellulose is a little unusual because each glucose tends to be bound more than just to the glucose in the chemical bond, covalent bond to one side and the glucose to the other side, but it also is hydrogen bonding. That's what the broken lines are, which are sort of yellowish or golden or brownish in color here. So this glucose here is bound to two other glucose covalently and bound to two other glucose by hydrogen bonds, meaning that cellulose has a bunch of hydrogen bonds holding the cellulose together. And cellulose, if you don't know, is a structural molecule in plants responsible for giving the plant its cell shape or its organism shape as well. Cellulose is a difficult molecule to dissolve because there are so many hydrogen bonds holding it together. The cow, in fact, cannot digest cellulose directly. What the cow does is it has a bunch of microbes in its stomach which digest the cellulose. And to be a little bit more complex, it has a protist in its stomach, and the protist has bacteria in the protist stomach, and it's the bacteria in the protist, which is in the cow's stomach, which digest the cellulose. And I know that's complex, but we'll just say microbes in the cow's stomach digest the cellulose. And that's why the cow can eat grass, which has a lot of cellulose. Humans do not have the bacteria in our stomach to digest grass. So if you were to eat grass, it wouldn't give you much, if anything, for digesting. Any question about any of that? We'll talk more about peptidoglycan when we talk about the cell wall. Just now, you need to know that peptidoglycan is at least partly a carbohydrate, and that's the come on, the ring structures here and here hooked together like that. And we'll talk a little bit more about peptidoglycan later in the class. I don't think we talk about it today. All right, any questions about glucose and carbohydrates? If not, let's begin talking about lipids. Lipids consist of carbon, hydrogen, and oxygen. And although they have oxygen, they do not have much oxygen. Let me show you in the next slide. So here is a lipid. Let's see if I can blow this up. And this lipid does have oxygen right there and right there. And this will actually come off when this binds to something over here. But the point is, it does have some oxygen, but not very much. Most of the atoms are carbon and hydrogen in a lipid. Lipids are generally nonpolar and insoluble in water. We call them hydrophobic. I'm saying generally because there is one exception, and we'll talk about that. Lipids are structural and energy storage molecules for cells. And lipids are a primary component of the cell membrane. We'll talk a little bit more about that, but the membranes are mostly made up of lipids. There are 3 different types of lipids: a simple lipid, a complex lipid, and a steroid. Fatty acids are a simple lipid, and they are a major component of many bigger or complex lipids. Fatty acids consist of a chain of carbons, so they contain mostly carbon and hydrogen. Most of the carbons contain a hydrogen or 3 or 2. There is a carboxyl group at one end, COOH, but that's it. That's the only oxygens in the fatty acids. You can have 2 types of fatty acids: a saturated fatty acid, which is what is shown here. A saturated fatty acid has each of the carbons held together in the fatty acid by a single carbon bond. And then you can have an unsaturated fatty acid where 2 or more of the carbons are held together by a single double carbon bond. Now, let me go ahead and show you that. See if I can blow this up. So this fatty acid, there we go. This fatty acid is a saturated fatty acid because it only has single carbon bonds between the carbons. And then this fatty acid here has a double carbon bond. So this is an unsaturated fatty acid. You only need 1 double carbon bond to become an unsaturated fatty acid. Any question about any of that? Let me see if I finished the last slide. Wherever you have a unsaturated fatty acid link, meaning wherever you have the double carbon bond, it'll put a kink in the chain, in the fatty acid chain. And you can see where we have the double carbon bond, it's no longer straight like this. We have a kink right there. So a simple lipid is also called a fat, and triglycerides are the most common fat in the body. Fats are also called triglycerides. Fats contain 3 fatty acids bound to a glycerol. Here is glycerol, and there's the fatty acid. And how you make that molecule is you pull out an OH group from the carboxyl group and a hydrogen from the glycerol group. And then you bind this O directly to this carbon to make the fat. A triglyceride has 3 fatty acid groups bound to the glycerol. And that's as many fatty acids as glycerol can bind to. For this molecule to be an unsaturated fat, all it needs is 1 double carbon bond. So this is a unsaturated fat, even though this and that carbon link is only a saturated fat. So this one, this hydrocarbon chain of the fatty acid group is saturated, and this one is saturated, but all you need is 1 in the entire molecule to call it an unsaturated fat. Triglycerides are when you have 3 fatty acids bound to a glycerol, and triglycerides are the body's richest energy source. And to see triglycerides, all you need to do is look down at your belly, and some of that fat you have down on your belly will be triglycerides. And it doesn't matter how thin you are, like I'm very skinny. I do have some triglycerides in my belly. So if you look down at your belly, you'll find some fat, and that'll be triglycerides. Now, I mentioned that the body's richest energy source is fats. So if ever you were like in Point Barrow, Alaska in the wintertime, and your car breaks down and you have some food in your car, and you have like 20 miles to get into the town or the nearest house where it's nice and warm, and you want to carry as much food as you can to give you energy so that you can keep warm. Let's say you don't have a very thick coat, so you need food to keep warm. The best food to take would be a fat, because you can get the most energy out of a fat. Per gram of fat, you'll get more energy out of it than a gram of protein or a gram of carbohydrate. Now, notice I'm saying that that would be what you want to do if you wanted to take as much food-bearing energy with you, meaning take fat, because you'll get the most energy out of it. However, I'm not saying that's the best diet. If you wanted to eat a good diet, you don't want to have a bunch of fats in your diet, especially unsaturated fats. Obviously, saturated fats are good to eat. Any question about any of that? All right, let's talk about the one exception. There is one lipid which is not hydrophobic. In fact, it's both, or it is a molecule that is both hydrophilic and hydrophobic. And that molecule is a phospholipid, and it's the main molecule in your cell membrane and all your other membranes of the cell, like the mitochondrial membrane. Phospholipid is shown here. It does have, let me blow this up. It does have two fatty acid chains bound to the glycerol. So there's the glycerol right there. And for humans, you're always going to have one saturated fatty acid and one unsaturated fatty acid, but this will depend on the species and the temperature that the species is exposed to, meaning I think animals that live in the Arctic tend to have more saturated fatty acids, and then animals that live in the tropics tend to have more saturated fatty acids. You don't need to know that. But the third functional group, there we go, attached to the glycerol is not a fatty acid. It's actually a phosphate group. And the phosphate group has PO4 with a negative charge. And then it has an R group attached to it. And R can stand for any organic grouping of molecules. And so we're not going to even state what R is. I could go look up what is the main R group in humans, but I don't know what it is. And unless somebody wants me to look online, I'm not going to look. But the point is, it's just an organic group. The phosphate linking to the glycerol gives this molecule different properties. Mainly, you'll note the phosphate is ionic, and then this part of the molecule, which is called the head, and that's the glycerol part, and the phosphate part is actually hydrophilic. So this part of the molecule loves to dissolve in water. this part of the molecule, the fatty acid portion, is hydrophobic, and it does not like to dissolve in water. It'll be repulsed from water. And this part of the molecule is wanting to dissolve in oil or some other liquid lipid or some component that would dissolve in oil. I used to know a chemical that lipids would dissolve in, like oil, but I don't remember what it is. It's some chemical I used as an undergraduate in my chemistry class, and I just don't remember. The point is that the phospholipid is both polar on this end of the molecule and nonpolar on this end of the molecule. Any question about that? This end of the molecule we call the head of the phospholipid with the phosphate group, and then the fatty acid tails are called the tail region of the phospholipid. So if we can draw this molecule that way, showing it in stick figure, and then when you put all of these molecules together in water, you'll start to get this forming. And the reason is that the heads like to be around the water. The tails do not like to be around the water. The tails like to be around other tails. So the tails will all come together. And this is actually what our cell membrane is made up of. Mainly phospholipids, making a lipid bilayer of our cell membrane. And it is true in animals, our membranes tend to have cholesterol, that's what that's showing you, in the membrane, as shown here. And the cholesterol gives this membrane added strength and sort of resilience. So the cholesterol makes the membrane stronger. And once you add the phospholipids all together in the water, you'll first get them to forming that, and then you'll get them to form this, where the outside of the liposome, for example, will have the phosphate heads facing the water on the outside, and then the tails will all be on the inside, totally away from the water, and with another tail because they're hydrophobic. And then on the other side, we'll have the heads, and then we'll have water inside the liposome. What does this look like? It should remind you of something. What has mostly water outside of it and mostly water inside of it, and a lipid bilayer forming like this. Nobody's going to guess? It doesn't look like anything to you? It looks pretty much like a red blood cell to me. And most of our cells, as well as most bacteria cells, will have the cell membrane with heads facing inside the cell, because the cytoplasm inside the cell is mostly water. And so you want the heads facing the mostly water. And then outside the cell will also mostly be water. Think about a red blood cell in blood. It's going to be in the blood. The blood is mostly water. So the heads are facing outside the membrane so that the heads can face the water. Any question about any of that? All right, if there's no questions, I think I'm going to end here. Let me make sure we end at 6:20. Yes, we do. And then I'll start here next time. Any questions before I go? All right, not hearing anything. I'll see you at 6:30 for the lab.